Have you ever noticed the alarming rise in conditions like obesity, asthma, type 1 diabetes, and, gosh, even severe food allergies? It truly feels like a new kind of modern plague is sweeping across the developed world, and it seems to be impacting people, even children, at younger and younger ages. It's hard not to wonder, what on earth is going on?
It's a trend that's definitely concerning and deeply perplexing for many
Right.
people because these aren't isolated issues, right? They really do seem to be interconnected somehow.
And in this deep dive, we're going to unpack a really groundbreaking idea. It suggests that these seemingly disparate modern health crises might actually share a surprising common link. It's the quiet, often unseen destruction and disappearance of vital, beneficial bacteria that have coexisted with humans for, well, millennia. We'll explore how some modern practices, things intended to save lives, make us healthier, might have inadvertently created a whole new complex set of challenges.
Yeah. Get ready to explore an unseen world inside and all around us. It's a world so profound it might just redefine how you think about health, immunity, and maybe even what it means to be human.
Okay, let's unpack this. To really get what's happening, we need to zoom out, way out, and appreciate the sheer, overwhelming scale of microbial life on Earth. Most of us think of Earth as a planet of plants and animals, but that's not quite the full picture, is it?
Not at all. Earth is fundamentally a microbial planet. For about 3 billion years, bacteria were the sole inhabitants. They were driving all of the essential chemical reactions that literally made the planet habitable for everything else that came later.
3 billion years, that's just staggering.
It really is. And to give you a sense of their dominance, imagine the entire tree of life as this huge, sprawling forest.
Mhm.
Humans, and maybe corn, let's say, would be just two tiny neighboring seedlings. You could barely tell them apart from a distance. The rest of that entire immense forest is almost entirely bacteria. Their diversity is just vast.
Wow. So if we're just two little saplings in this enormous bacterial forest, it really highlights how fundamental these organisms are. What exactly are these essential, maybe world-shaping roles that microbes perform for the planet?
Oh, their work is absolutely critical. They created the oxygen we breathe, they form the very soil we farm, they decompose matter, endlessly recycling nutrients. They even fix inert nitrogen from the atmosphere into forms that all plants and animals need to survive.
So, like, without them, life as we know it just wouldn't exist.
Pretty much, yeah. And they do amazing things. Think about the Deepwater Horizon oil spill. Bacteria actually ate up a lot of that contamination. They combine nutrients in the oil with nitrogen from the air, like a natural cleanup crew.
That's incredible. And they live in extreme places too, right? Places we wouldn't think anything could survive.
Absolutely. We're still barely scratching the surface. They thrive in hydrothermal vents with boiling water and toxic chemicals, super high geysers under massive Antarctic glaciers, the Marianas Trench, the deepest place on Earth, active microbial community down there.
Seriously,
Yeah.
way down there.
Yep. They're even lofted by winds into the atmosphere, helping form clouds and ice for snow.
Yeah.
And get this, in South Africa's Imponing gold mine, bacteria survive using radioactive decay for energy. They can even mine gold, converting dissolved, toxic gold into solid, inert crystals, gold deposits.
Okay, mind blown. They're clearly foundational to the whole planet. But is it all just competition down there? Or is there cooperation too? We always hear about survival of the fittest, but that feels incomplete from microbes.
It's definitely not the whole story. Darwinian competition is there for sure. But the story of microbes is also just a saga of endless cooperation, synergy. For example, in your gut, one Bacteroides bacterium might detoxify a chemical that inhibits E. coli. So E. coli benefits, even if the Bacteroid doesn't get anything directly. That's called a commensal relationship.
Okay. And then there's true symbiosis, where both benefit, like a partnership.
Exactly. Imagine if E. coli's waste product is actually good food for Bacteroids. They'll tend to hang out together, helping each other. This cooperation goes way back. There are 3.5 billion-year-old fossilized microbial mats in Australia, different layers of bacteria doing different jobs, benefiting the whole community. And some microbes form these protective biofilms, kind of like a gelatinous shield against harsh environments, or even our own immune system.
So this intricate dance of microbial life, it's not just out there in gold mines or oceans. It's right here, inside us, our personal microbiome.
That's exactly right. Every creature, from starfish to humans, has co-evolved with its own unique collection of microbes. They carry out essential metabolic and protective functions for us. In humans, this includes diverse populations in our nose, where Staphylococcus aureus, the one often linked to infections, it can actually live benignly in maybe a third of us.
Really? Staph just hanging out.
Yeah. And our mouths are teeming with different bacteria on different surfaces. Like the gingival crevice between your teeth and gums. It's as dense with bacteria as your colon. It's why your breath smells different in the morning. Those anaerobic bacteria thrive when there's less air exchange while you sleep.
Okay, that explains a lot. So their influence goes beyond just living in us. They actively shape our physiology. That's almost hard to wrap your head around. How profoundly do they actually influence us?
It's fascinating just how deep it goes. They metabolize drugs we take, like digoxin for heart conditions. They actually affect how much of the drug reaches your bloodstream. They protect the vagina mac. Lactobacillus species produce lactic acid, making it acidic and hostile to pathogens. And maybe most importantly, your microbes act as a crucial, kind of like a third arm of your immune system.
A third arm? How so?
Well, they help form the immune system's memory cells, and they produce Treg cells. Think of Tregs as the immune system's calm-down crew. They prevent overreactions, making sure our defenses don't mistakenly attack things like pollen or food or even our own tissues.
So these microscopic residents are actually teaching our immune system how to behave, how to tell friend from foe.
Exactly. Our microbial differences, the variations between your microbiome and mine, are actually far greater than our human genetic differences. The specific mix you have is deeply personal. It influences your health, your metabolism, your immunity, maybe even your cognition.
Which brings us to this powerful concept, the keystone species. You hear about wolves in Yellowstone National Park. They're a classic example, right? When they were removed, the whole ecosystem went haywire.
It did. The elk population exploded. They ate all the willows, which impacted songbirds, beavers. It cascaded through the whole system. Ravens, eagles, coyotes, bison, all affected. And this concept applies directly to our microbiome. Remember that tiny stomach-dwelling bacterium, Helicobacter pylori?
Vaguely, yeah. Wasn't it linked to ulcers?
It was, but its loss, as we'll discuss, might be a profound cautionary tale, like removing the wolves. The loss of certain key microbial species can have these huge cascading negative effects on our health. H. pylori might be one of those microbial wolves.
Okay, so to really grasp the shift we're talking about, this loss of microbes, we need to step back in time to a world very different from ours. A world where infectious diseases were just rampant, a constant terrifying threat. You mentioned your father lost two sisters.
Yes, I never knew them.
Yeah.
In the early 20th century, they both died before their second birthdays, high fevers, and that was tragically common back then.
That's hard to even imagine.
Yeah, in 1850, as many as 4 in 10 English babies died before their first birthday. Lethal epidemics, cholera, smallpox, diphtheria, whooping cough, tuberculosis, they just swept through crowded cities. Child mortality was incredibly high. The Black Death, caused by Yersinia pestis from fleas on rats, wiped out maybe a quarter to a third of Europe's population in just a decade. Disease was this ever-present shadow.
So what started to turn the tide? What gave us a fighting chance?
Well, the germ theory revolution was pivotal. You had Louis Pasteur in 1857 showing microbes cause fermentation and disease. Ignaz Semmelweis realized hand washing was key, dramatically reducing deaths in maternity wards. Joseph Lister pioneered antiseptic surgery using carbolic acid, and Robert Koch developed criteria, Koch's postulates, for linking specific microbes to specific diseases. This led to better sanitation, better understanding, but not yet widespread effective treatment.
They were looking for magic bullets, right? Drugs that could kill the germs without killing the patient. And then came penicillin, sort of an accidental miracle.
A truly serendipitous discovery, yeah. August 1928, Alexander Fleming returns from vacation, finds this Petri dish he'd forgotten to toss. It had a patch of blue-green mold, Penicillium notatum, and around it, the Staphylococcus bacteria just weren't growing. Something in the mold was killing them.
But it took a while to actually turn that observation into a usable drug, didn't it?
It did. The initial challenge was producing enough of it. The real breakthrough came during World War II. Scientists in Peoria, Illinois, found a moldy cantaloupe of all things that produced huge amounts of penicillin.
A cantaloupe? Seriously?
Seriously. That strain, combined with industrial fermentation techniques, really scaled things up. Companies like Charles Pfizer and Company used things like molasses to grow these potent penicillium molds in huge vats. By D-Day, June 1944, they were making 100 billion units a month. It transformed battlefield medicine. Soldiers were surviving wounds that would have been fatal just years before.
So penicillin really ushered in this golden age in medicine, a true wonder drug. It must have allowed doctors to do things they could only have dreamed of before.
Oh, it was miraculous. Surgery became dramatically safer. Think about it. Open heart surgery, organ transplants, even IVF, none of that would really be feasible without antibiotics to control infection risk. Chemotherapy for cancer often wipes out the immune system, making patients incredibly vulnerable to infection. Antibiotics made chemo survivable. In ancient diseases, syphilis was virtually eliminated from China in the 1950s through mass penicillin campaigns.
So how did these wonder drugs actually work? What was their secret weapon against bacteria?
They were like the atomic bomb for bacteria. They attacked fundamental vulnerabilities. Imagine a drug that could just rip apart a cell's outer wall, its skin, or jam up the machinery inside that makes proteins, or just stop it from dividing, from reproducing. That's the kind of ruthless efficiency they brought. Most worked in one of those three ways, hitting the cell wall, protein synthesis, or cell division. It fundamentally changed the playing field between humans and microbes.
But like the atomic bomb didn't end all wars, antibiotics didn't end the threat of bacteria. There was an unfulfilled promise there, wasn't there? A catch.
Exactly. The hope was for a panacea, a silver bullet to end all bacterial threats. But bacteria are masters of evolution. The arms race, based on inherent resistance mechanisms that bacteria already possessed, just continued, maybe even accelerated.
Right, which brings us directly to this overuse problem. How did we get from wonder drug to a situation potentially fueling these modern plagues? After World War II, this incredible optimism, antibiotics became widely available. And it seems like we just started using them for, well, everything.
That's pretty much it. The post-war euphoria was immense, but it led to this extravagant use, especially for things like common colds or coughs, upper respiratory tract infections, URIs. The vast majority of those, over 80%, are caused by viruses. Antibiotics do absolutely nothing against viruses, yet they were prescribed constantly.
Why? Why were doctors prescribing them so readily, even when they probably knew it was likely viral? Was there some kind of doctor's dilemma going on?
Absolutely. It's complex. First, it's genuinely hard sometimes to tell a viral infection from a bacterial one without quick, reliable tests, which often weren't available. Parents, understandably worried, often expected antibiotics. They wanted the doctor to do something. And critically, there was this deep-seated fear of rheumatic fever.
Rheumatic fever. What's the connection?
Rheumatic fever is a serious, though rare now, complication of untreated strep throat, which is bacterial. It can damage the heart valves. So doctors often prescribed antibiotics, not necessarily to treat the sore throat itself, but as a preventative measure against a small chance of rheumatic fever, even if they suspected the illness was viral. The thinking was often, well, the antibiotics probably won't hurt, and I want to cover all the bases just in case.
And this wasn't just occasional. This happened on a truly massive scale, right?
Ooh, staggering scale. In 2010 alone in the U.S., healthcare providers wrote 258 million courses of antibiotics. That's about 833 prescriptions for every 1,000 people. Pediatricians were major prescribers, maybe 25 million courses a year just from them. Even dentists contributed significantly.
Right.
It was like standard practice for almost any sniffle or procedure.
And this widespread, often unnecessary exposure, it inevitably led to the rise of antibiotic resistance. It's like natural selection on hyperdrive, isn't it?
Precisely. Every time you expose a large bacterial population to an antibiotic, you kill the susceptible ones.
Yeah.
But any bacterium with a random mutation that confers resistance survives. It's then free to multiply, passing that resistance gene along. We essentially selected for resistance on a massive scale. And we've seen the devastating human cost. You hear these stories. Brandon Noble, a pro football player, his career ended by MRSA methicillin-resistant Staph aureus after routine knee surgery.
Just awful. And it's not just elite athletes or hospital patients anymore, is it? This threat is moving out into the community.
That's the truly chilling part, yes. Ricky Linetti, a college football player, died from an MRSA abscess that started as just a small sore. A congressman's friend died from MRSA after a knee replacement. These aren't isolated hospital infections anymore. Resistance is out there, affecting otherwise healthy people in everyday life.
And it's not just about us taking antibiotics directly. There's this whole other dimension. Antibiotics on the farm. That seems like a huge, often overlooked contributor.
A massive one. For decades, many conventional farms, especially in the U.S., have used subtherapeutic, meaning low, non-treatment doses of antibiotics in livestock feed. not primarily to treat sick animals, but to promote faster growth and prevent infections in crowded feedlot conditions. This practice was actually banned in Sweden back in 1986 and across the EU in 1999 because of resistance concerns.
Right. And the evidence clearly shows these resistant microbes from animals do make their way to humans, despite what some industry groups might argue.
Yes, absolutely. Despite denials from some food producers and pharmaceutical companies, the evidence is overwhelming and goes back decades. Studies found identical strains of antibiotic-resistant salmonella in sick people and in the animals that had been fed those growth-promoting antibiotics. A Danish study in 2013 used whole genome sequencing, super precise genetic fingerprinting to show that MRSA infections in 2 farmers came directly from the same strain infecting their pigs. The link is undeniable.
And we're not just exposed through eating meat or direct contact, are we? These drugs seem to permeate the environment.
That's right. The antibiotics themselves and the resistant bacteria end up in the environment. They leave residues in food products like milk, meat, eggs. They get into waterways through farm runoff and even through treated human sewage because our bodies don't fully break them down. A 2009 study found antibiotic-resistant bacteria and resistance genes in source water, in treated drinking water from plants, and even in tap water in several U.S. cities. It creates this constant low-level exposure, keeping the pressure on for resistance to develop and spread.
So is this resistance problem entirely new, something we created from scratch? Or are we just amplifying something ancient?
That's a really critical distinction. Antibiotic resistance itself isn't new. The genes for it existed in bacteria long before we discovered antibiotics. Scientists found resistance genes in bacteria frozen in 30,000-year-old permafrost. Microbes have been fighting each other with chemical weapons for billions of years, but human activity, primarily our massive overuse of antibiotics in medicine and agriculture, has amplified the prevalence of resistance by, well, many orders of magnitude, as the book puts it. We've created this intense global selective pressure that never existed before, so we can't ever fully make resistance go away because the potential is always there, but we've turbocharged the process dramatically.
Okay, this leads us to what happens even earlier in life, the section on early life interventions and how they might be, wow, literally rewiring human development. You mentioned historical examples like thalidomide and DES,
Yep.
powerful lessons about unintended consequences, especially for babies.
They really are powerful, tragic, cautionary tales. Thalidomide, given for morning sickness in the late 50s, early 60s, led to thousands of babies born with severe limb defects, a disaster. And DES, diethylstilbestrol, an estrogen given to pregnant women supposedly to prevent miscarriage. It devastating affects infertility, higher rates of preterm birth, a rare vaginal cancer in the daughters exposed in utero didn't show up for decades. It just highlights how critical that developmental window is and how interventions can have long-term, unforeseen consequences we just didn't anticipate.
And a major, almost silent intervention happening constantly today is the rise in C-sections. You called it the C-section epidemic. The rates have really soared, haven't they?
They really have, globally. In the UK, it went from maybe 1 in 10 births in 1980 to 1 in 4 by 2008. Brazil's rate is nearly 50%. Some hospitals in Rome report rates as high as 80%. Compare that to Sweden, where it's around 4%. This isn't just about surgical trends. It has profound biological implications for the baby's first encounter with the microbial world.
Because it completely bypasses that crucial microbial handoff during vaginal birth, that first sort of signature splash of microbes.
Exactly. A baby born vaginally gets coated, inside and out, with their mother's vaginal microbes, mainly species like lactobacillus. This is likely what evolution intended the baby's first inoculation, its first introduction to its microbial partners. C-section babies, however, their first colonists are typically skin microbes from the mother, the father, the doctors, the nurses, like Staphylococcus, Corynebacterium, Propionibacterium, plus whatever's floating around the operating room. Their founding microbial community bears no resemblance to their mother's vaginal flora. And while their microbiomes might converge somewhat later in infancy, those very first signals to the developing immune system and metabolism could be critically different.
And it's not just the mode of birth. Antibiotics given during pregnancy or to newborns are also incredibly common. What are the ripple effects there?
Yes, very common. Pregnant women often get antibiotics for urinary tract infections or other minor things. And a big one is preventative antibiotics, given during labor if the mother tests positive for group B strep bacteria. But the statistic is pretty stark. You have to treat around 400 women with antibiotics during labor to prevent just one serious group B strep infection in a newborn. That means hundreds of mothers and babies are being exposed, potentially skewing the mother's microbiome right before birth, and thus the baby's initial colonization, even before they're born. And while these drugs prevent some GBS infections, they might inadvertently select for other resistant bacteria, like certain E. coli strains, potentially increasing the risk of other types of infections later. It's a trade-off with hidden costs.
Wow, it feels like all these factors are converging, a perfect storm, almost. Let's look at some of the direct evidence linking these microbial disruptions to specific modern diseases, starting with obesity.
Right. There's fascinating work. Ruth Lee studied germ-free mice raised with no microbes at all. When she transferred gut microbes from women in their third trimester of pregnancy into these mice, the mice got fatter and had a higher blood sugar compared to mice given microbes from first trimester women. Suggests the late pregnancy microbiome is geared towards energy extraction.
So the microbes themselves influence the mice's metabolism.
Exactly. Other studies directly show that giving mice antibiotics early in life led to them developing more body fat later on. And in humans, large epidemiological studies, like the ALS-PAX study in Britain, found links between both C-section birth and antibiotic use in early infancy and a higher risk of childhood obesity. It strongly suggests that the composition of our microbiome, especially early on, plays a direct role in programming our metabolic development.
So it's not just about calories in, calories out. It's also about who's processing those calories inside us. What about asthma and allergies? They seem to be absolutely skyrocketing.
Yeah, and here that keystone species, Helicobacter pylori, comes back into the picture. Remember, the stomach bug. Multiple studies have found an inverse relationship between having H. pylori in your stomach and the risk of developing asthma and allergies, particularly childhood onset asthma.
Inverse. So having it protects you, that seems counterintuitive for a bacteria linked to ulcers.
It does, but the thinking has evolved. Especially certain strains of H. pylori, the ones called Kaga-positive, seem particularly good at interacting with our immune system. They almost act like microbial diplomats, communicating with our immune cells and encouraging the production of those T regulatory cells we talked about, the ones that prevent overreactions. By taming the immune system early in life, H. pylori might help prevent it from freaking out over harmless things like pollen, dust mites, or certain foods. Its disappearance from many people in developed countries could be removing that protective influence.
I remember you mentioned your daughter Genia and her story. It seemed to connect here.
Yes, personally. Jeannie had quite a few courses of antibiotics as a young child for recurrent ear infections. Later, she got metronidazole for a Giardia parasite infection. Afterwards, she developed symptoms very much like celiac disease and also a sudden severe allergy to mangoes. And intriguingly, large Swedish studies have shown about a 40% increased risk of developing celiac disease if you've had prior antibiotic use, particularly with metronidazole, which is known to really disrupt gut bacteria. It feels plausible, at least, that these exposures wiped out microbes that were providing some kind of protective allergy-suppressing function.
Wow. And type 1 diabetes, another autoimmune disease, sadly on the rise in kids. Is there a microbial connection being explored there too?
Yes. Research, again, often starting in mice models, shows that giving antibiotics after birth can accelerate the onset of type 1 diabetes. And critically, these studies found changes in the immune cells within the intestines even before any damage showed up in the pancreas where insulin is made. This suggests it might be a gut-first problem. Something goes wrong with the microbial immune interaction in the gut, and that triggers the autoimmune attack on the pancreas later.
It's such a complex web. Are there even theoretical links being drawn to things like autism or hormone regulation? The idea that gut bugs could influence brain development or hormones sounds almost like science fiction.
Well, maybe not quite sci-fi anymore. Consider this. Your gut contains over 100 million neurons. It's often called the second brain. This enteric nervous system communicates directly with your main brain via the vagus nerve, influencing things like mood and cognitive function. And guess who's constantly interacting with those gut neurons? Your gut microbes. They produce neuroactive chemicals, including things like gangliosides that the developing brain actually needs. So disrupting that microbial community, especially early on, could plausibly impact complex brain development. It's an active area of research.
And hormones.
Yes, that too. A classic example is women on birth control pills sometimes experiencing breakthrough bleeding if they take certain antibiotics. Why? Because gut microbes help metabolize the estrogen from the pill. Wipe out those microbes, and hormone levels can fluctuate unexpectedly. It shows a direct link between gut bacteria and hormone processing. It's all interconnected.
It really seems like we keep circling back to this central idea, the compounding effect. These early life exposures, C-sections, antibiotics aren't just isolated events. They add up generationally, perhaps.
That's the core thesis, yes. that many of these diseases that have dramatically increased since the mid to late 20th century, obesity, asthma, allergies, autoimmune conditions, might be fueled by this single overarching factor. It's the profound change, the depletion of our ancient resident microbial communities, especially during those critical developmental windows in early life. And the effects might be compounding across generations. Each generation starts with a slightly less diverse, slightly less robust microbiome, making them potentially more vulnerable. It's like our collective microbial bank account of defenses is getting depleted.
That sounds genuinely alarming. It paints a picture of an antibiotic winter, echoing Rachel Carson's silent spring about pesticides. What are the dangers ahead if we just keep going like this? What's the hidden cost we're not fully appreciating?
The pine beetle metaphor used in the book is quite apt, I think. Pathogens, like pine beetles, are always around. But when the forest, our microbial ecosystem, is weakened, stressed, or missing key players, it becomes much more vulnerable to attack. Experiments have shown that even a single dose of some antibiotics can increase susceptibility to infection by pathogens like Salmonella by, get this, 30,000-fold.
It's 30,000.
30,000-fold. Because the antibiotic wipes out the protective resident microbes that normally keep pathogens in check. Antibiotics don't just kill bad bugs, they destabilize the entire microbial community, sometimes for long periods. This is the hidden cost. Increased vulnerability. It's why we see things like deadly C. diff infections surging after antibiotic use, like in the tragic case of Peggy Lillis mentioned in the book, who died after taking an antibiotic for a minor dental procedure. And it's why antibiotic-resistant threats like MRSA are spreading from hospitals out into the wider community. Our depleted defenses are making us easier targets, potentially fueling future epidemics.
Okay, so this is serious, but the book doesn't just leave us there. It talks about moving towards an antibiotic spring. How do we even start? It feels like turning a huge ship.
It is a huge ship, but not an impossible one to turn. It starts with fundamentally rethinking medical practice. Doctors need to become much more cautious, moving away from just reflexively prescribing antibiotics towards really careful judgment. Is this truly bacterial? Is this antibiotic necessary? What are the potential long-term costs to the microbiome? There might even be a shift in liability one day from doctors fearing being sued for not treating with antibiotics to potentially being sued for the harms caused by unnecessary antibiotic treatment. That would be a major paradigm shift.
And what about getting smarter with the antibiotics we do use, more targeted approaches?
Precision medicine is absolutely crucial here. We desperately need to develop more narrow-spectrum antibiotics. Drugs that target only the specific pathogen causing the infection, leaving the beneficial bystander microbes unharmed as much as possible. But to use those effectively, we also need rapid diagnostic tests that can tell a doctor right there in the clinic within minutes, yes, this is bacterial, and it's this specific bacterium susceptible to this specific drug. We need tests based perhaps on chemical signatures of infection or the host's immune response.
That makes perfect sense. Is that happening?
The science is progressing, but there's a big economic hurdle. Pharmaceutical companies, frankly, make more money from broad-spectrum antibiotics that can be sold for many different types of infections.
Yeah.
Developing and marketing narrow-spectrum drugs and the companion diagnostics is currently less profitable. It's a market failure we somehow need to address, maybe through public funding or incentives.
Okay, so better stewardship and smarter drugs are key. But what about actually restoring what we've lost? This idea of microbial restoration seems really exciting, almost futuristic.
Yes, and this is where a lot of the hope lies for the future. Let's start with birth. Remember C-sections bypassing the vaginal microbes. There's growing interest in vaginal seeding. It sounds a bit strange, but it involves taking a gauze swab of the mother's vaginal fluids shortly before a planned C-section, and then wiping the baby's mouth, face, and body with that swab shortly after birth, trying to mimic the natural exposure.
And does it work? Does it change the baby's microbiome?
Early studies, including pioneering work by my wife Gloria Dominguez-Bello, suggest, yes, it does shift the C-section baby's microbiome to look more like that of a vaginally born baby, at least initially. More research is needed on long-term health outcomes, but it's a promising low-tech approach. Gloria's work also compared the microbiomes of Amerindian villagers in the Amazon who'd had virtually no contact with modern medicine or antibiotics. They're like living fossils of microbial diversity with modern Americans. The differences were stunning. The villagers had vastly greater diversity, including over 100 unique bacterial species not found in most U.S. subjects. It starkly shows what we've likely lost.
Wow. So seeding is one approach. What about more personalized restoration later in infancy?
I really envision a future, maybe not too far off, where at a baby's 1-month or 6-month checkup, we analyze their stool and urine, We could identify specific beneficial microbes that seem to be missing or are present in very low levels compared to healthy babies, maybe key bifidobacterium species, or Allobaculum, or Oxalobacter, which helps prevent kidney stones. And then the pediatrician could prescribe a personalized culture of just those specific missing bacteria, a targeted microbial top-up.
Like a truly personalized probiotic tailored exactly to that baby's needs. That sounds revolutionary. And we already have a form of microbial restoration being used now, right? Fecal microbiota transplants, FMTs.
Yes, FMT has been a game changer for treating severe recurrent Clostridium difficile infections, which often happen after antibiotics wipe out the normal gut flora. Taking stool from a healthy donor and transferring it to the sick patient can restore a healthy gut ecosystem and cure the C. diff infection with remarkable success rates, often over 90%.
And could FMT be used for other conditions linked to microbial disruption?
That's the big question and the focus of intense research. Could it help with inflammatory bowel disease, Crohn's, ulcerative colitis, celiac disease, irritable bowel syndrome, maybe even obesity, metabolic syndrome, allergies? There's even early-stage research looking at autism spectrum disorders, given the gut-brain connection. If a disordered gut microbiome is a root cause or major contributor to these conditions, then restoring it via FMT holds enormous therapeutic potential. But we need more rigorous trials.
What about the probiotics and prebiotics we see everywhere in supermarkets and pharmacies today? Do they fit into this picture?
Right now, the probiotic market is honestly a bit of a wild west. Lots of products, lots of claims, but often very little rigorous scientific evidence for specific health benefits in humans. Many are untested. That's not to say they're useless, but we need much more science. We need large, well-controlled clinical trials to figure out which specific strains at which doses actually help which conditions. Prebiotics, essentially food for beneficial bacteria, like specific fibers found in human milk or certain plants, are also important for nurturing the good bugs already there. And symbiotics combine probiotics and prebiotics. The potential is there, but it needs to move beyond marketing hype into solid science.
And maybe the most profound idea, the one you predicted years ago, actually reintroducing specific disappeared organisms back into our population, like H. pylori.
Yes. It might sound radical, but if we've lost key microbial partners that co-evolved with us for millennia to perform vital functions like regulating our immune system, then maybe the ultimate solution is to carefully, scientifically give them back, especially to children during that critical window of immune development. The idea I put forth in 1998 was that we might one day be selectively giving beneficial strains of H. pylori back to children who lack it, to potentially restore protection against allergies and asthma. The scientific evidence supporting this concept has only grown stronger since then. It's about identifying those crucial missing pieces of our ancestral microbiome and finding safe, effective ways to restore them, filling that modern microbial void.
What a monumental thought, to actively reverse this decades-long trend that's been shaping our health in such fundamental ways. It brings us right back to that starting point. Our incredible medical innovations, antibiotics chief among them, have saved countless lives. But they've had these unforeseen, massive consequences for our internal microecology. The scale of impact, as the book suggests, is almost as hard to grasp as, say, the melting ice caps resulting from burning fossil fuels, an unintended consequence of progress.
That's a powerful analogy, and I think it holds true. The impact has been enormous, largely invisible and deeply consequential. But, and this is important, there is real cause for optimism. These dramatic shifts in our microbiome, this rise of modern plagues, they've happened incredibly fast, mostly in the last 60 or 70 years. It's a blink of an eye in evolutionary time, even in human history. Change that comes that fast can potentially be reversed just as rapidly, or at least managed and mitigated now that we understand the problem in a way we never did before. The science is exploding.
So we really stand at a crossroads. We have these powerful tools, these practices that have served us well in many ways, but the unintended consequences are now becoming clear. It makes you think, what will you, listening now, consider differently about health choices, especially for your children, knowing the profound impact of this unseen microbial world?
I truly believe that by consciously working to understand, protect, and maybe even restore our vital missing microbes, we can do more than just treat symptoms. We might be able to actually reverse the tide of these modern plagues, paving the way for a much healthier future for generations to come. We can transform this potential antibiotic winter into a flourishing, more balanced antibiotic spring.